Our next talk and last talk of the day is by Randy Yurden, who's the founder and CEO of BioSpherix and BioSpherix Medical, and he's going to talk about his really unique technology for GMP processing.
Thank you, Kyle, and thank you for a few minutes here to share a few ideas that might be helpful. The title of my talk is directed toward therapeutic cells, but the concepts I want to talk about are pertinent for any type of cell. And in the context of cell therapy, obviously the goal is to produce a very high quality cell, the highest quality possible. But what is a high quality cell? Well, first of all, it probably is not highly variable. Second of all, it's probably very robust. And third, it's probably very phenotypically intact. And of course, fourth, probably, its genotype is also very intact. So the idea of a high quality cell is the essence of what I want to talk about today. And there's many factors that contribute to a high quality cell, but some of the most fundamental universal factors are listed here. These critical process parameters that we all know and love. Temperature and CO2 and O2 are the ones that are provided by the environment, and of course the other ones are secondary to those. And I use this slide for two reasons. Number one, to maybe get your attention, and number two, to illustrate a real critical, amazing capability that the body has for the cells inside of it. It has an incredible ability to hold these critical process parameters very tight over long periods of time. And if we could only do so well when we take them out of the body, I think we'd be a lot further along than we are now. But that's basically what I want to talk to you a little bit about, and hopefully give you some insight into maybe improving things. And I'm using the term of suboptimal transients to illustrate a real simple point. Is when you have optimal conditions, and I'm just going to deal with these simple little critical process parameters. There's many others, obviously, but these are always involved. When they're not in optimum levels, it's suboptimal. And I'm going to call them transients, because the way that we think about cells in the dish or cells out of the body is that normally they're in an incubation state. They're either in an incubator or a bioreactor where these critical process parameters are pretty much optimized. And I think the assumption that we all make is that since they're in optimal conditions most of the time, that's probably good enough. And what I'm going to show you is that's not the case. And basically, we've been 100 years dealing with these suboptimal transients. It's over 100 years, if this information is correct, when cells first were put into the dish successfully. And this represents the relentless march of this industry, the relentless march of technology, and it shows how many different aspects of propagating, growing cells in the dish or outside the body has improved. There's many different culture vessels. There's all sorts of different culture medias and additives, and there's been a lot of advancements, and these advancements are occurring every day, all the time. But in the environment where we grow and handle and incubate cells, there's really been very little advancement. And the incubation environment, I would say, has a little bit of effort here, but the handling environment, the machine environment where the transport, none of that has really been attended to. So just to kind of summarize, a little simplification is when cells are outside of the body and they're still metabolizing, and I say that to exclude in the freezer, they're really, they're only in 4 places. They're either in an incubation state, whether it's in a conventional incubator or one of, any type of a bioreactor, or they're being handled, usually in the hood, or they're in some kind of a machine. And there's many different types of machines now, analytic process machines for doing cells. And then finally, they're in transport between 2 of these. So this is a simplification, and I'd like to address each one of these really quickly. This is the only place right now where we tend much attention toward these critical process parameters. Yet look at an incubator in a busy lab or a busy clean room where they're putting cells in and taking them out every every hour or so. You see lots of excursions to suboptimal levels in all 4 of these critical process parameters. And this is just an actual measurement to show a lack of control with the O2 and CO2 in a typical incubator. Bioreactors are much better because they're actively controlling, and they are presumed to be better able to keep optimum conditions. But when you look at handling, it's always suboptimal. And the duration of these suboptimal conditions, can vary quite a bit depending on many factors. Machines, usually the same thing. There's a huge, consequence of the beneficial aspects of taking cells and putting them in machines is they experience suboptimal conditions. And this includes the upstream, staging, getting ready to go into the machine, maybe the downstream as it's waiting for other samples. So again, variability in these critical process parameters. Transport, same concept, usually suboptimal conditions. So what have we been doing for 100 years here? We've gotten quite a ways, and I think it's easy to, it kind of boils down to we're depending upon 2 factors that are real. One of them is media inertia. Media has a certain capability to hold critical process parameters for a period of time, and cells have an ability to respond in a defensive way to withstand these exposures to suboptimal conditions that we force them to endure. So, when a cell when a cell preparation is taken into a uncontrolled environment where there is a suboptimal condition that is transient, it's going to last a while, the first thing we depend upon is media inertia. And this does last, and in many cases, this is key to withstanding some of these short-term exposures to air. And once the media inertia, well, I should say that there's many factors that determine how long this takes place. And there are some clever culture vessel designers who have come up with a way to maximize the media inertia for the benefit of cells. But it varies depending upon different factors. But once the media starts to drift away from optimum conditions, the cells will recognize it. The cells will then start to respond in a homeostatic way to survive and to endure these suboptimal conditions. And they have incredible abilities, frankly, to withstand all sorts of different assaults, and they use all sorts of different mechanisms specific to the assaults. And once they get to the point where they can't endure these suboptimal conditions anymore, what happens? They start to die. The viability starts to drop off. And this is one of our critical basic metrics that we use to evaluate the quality of our cell populations and so on. And this loss of life is a legitimate way to monitor how well we're doing in the production of therapeutic cells or even in the production of experimental procedures. But this is crude. And what happens long before this is the cells will run out of energy. They basically have to survive. So they're gonna use the energy that it takes to survive. And if they have to go long enough because the optimal conditions aren't corrected, then they start to run out of energy, And at this point, what happens is you have a loss of vigor. Your cells become weak. They don't have a robust capability. And of course, if you're going to inject them into a patient to cure their disease, that's not a good thing. So you got to realize that this time frame is something that isn't normally looked at. Now, there's some new tools out there that allow you to measure this robustness, this energy store, that I would highly recommend, because it's going to give you better results than just viability. But another thing that's really important to understand, and it's kind of the main focus of what I want to talk about here, is long before the ATP runs out, what happens is the cells switch from survival mode, or from functional mode, which is what they were designed to do, to divert away from the phenotypic function that is normal for them, to survival. They suspend their phenotypic programs, their phenotypic functions, and they move over to a higher priority to survive. And this is something that comes quite a bit of time beforehand. And I think this is something that has to get considered in much more important ways than it does, because at this point, the cells are losing their function. They're losing their potency, and this is the new metric for therapeutic cells. So anytime you're not in optimal conditions currently, you're depending upon media inertia and cell homeostasis, and you're risking the robust capabilities of the cells and their potency. Cell manufacturing is kind of represented here, and it's important to kind of remember that a lot of cell manufacturing procedures go in and out of incubation or expansion states, and so it's really something to keep track of. A question that I bet 9 out of 10 experienced cell production people are asking is, yeah, but are they really that important? After all, we've been doing it this way for 100 years, and after all, my cells have been doing fine for 20 years. So what I'd like to do is try to convince you that they are important, and I'm going to use a couple examples. Now, this example is one we probably are all familiar with. Anybody who has been taught how to culture and handle cells understood from Biochemistry 101, just before they learned how to culture cells, is that pH affects the three-dimensional conformation of all macromolecules, all the ones on the surface of the cells, maybe some on the inside of the cells, and that can't be good if something's happening there. And they look in the incubator for the first time, and they see all that pretty orange color, and they ask what that? Well, that's the phenol red showing that the pH is right. And then they get shown how to take them out of the culture or the incubator to the hood, and then they notice it turning red, and they say, gee, that doesn't look too good. What's that mean? Well, that means it's the pH is changing because we don't have the CO2 anymore. And they say, oh my gosh, that doesn't sound too good. Isn't that hurting the cells? And the mentor always says the same thing. They say, no, it doesn't seem to hurt the cells. We've always been doing that way. It's fine. But the reality is everybody hurries in the hood because they intuitively know that it isn't good. And in the GMP world, it's usually required that you document the time in the hood. So the problem has to be real. Otherwise, these things wouldn't be happening. So the lesson here is science tells us this. And I would say we can listen to science. And beginners intuitively see it. And experienced people, they practice it. They try to minimize those negative consequences regardless of what they are. They don't know what they are yet. But they know they're intuitively that they're not good, and they hurry to minimize the consequence. And in GMP, and I'm going to say this a little cynically, GMP accepts the consequence, proceduralizes to achieve consistent negative consequence with no measure of consequence, and documents to confirm consistent negative consequence. My second example is with temperature. And this is a publication that came out a couple years ago. It came out of Bayer. And they were noticing some major variations in their huge production bioreactor on the amount of product that was being secreted. And this variation was troublesome to them. I think it was a coagulation factor. So they decided to do a systematic investigation and look all the way back through the process to see, where this variation was coming from. Obviously, big problem. And what they discovered was really amazing and really surprising. And they investigated all the way back, the entire seed train, where there was many assumptions that maybe it was coming from here, it probably was coming from there. Everything that they tested came to the conclusion that this was happening with one suboptimal drop in temperature from 37 down to 22. Well, think about that. How often do we do that with cells? And when they corrected this single suboptimal transient, the variation went away. So, there's some lessons to be learned here too. Just one suboptimal transient, just one, right? And who would have ever thought it would be temperature? Temperature is a pretty innocuous thing. Things cool down and slow down and they warm up and speed back up, but gee, it shouldn't do something that bad. And then the other thing that's really significant is it was the progeny of those cells. It wasn't the cells that actually experienced the suboptimal transient, it was the progeny, quite a few generations later. And, that's something that has to kind of get into consideration. Another significant thing to me is these were these freak immortal cells that are almost impervious to nothing. A Chinese hamster ovary tumor that was isolated 40 years ago and has been propagated everywhere all the time, that, how could that be affected? So it's not just these early passage cells we're using for cell therapy that we want to retain, these are, the very strange, unnatural cells that it's kind of surprising. Another significant lesson here is what was affected. It was the genetically engineered phenotype that was affected. It was the ability of this genetic construct to produce the protein and secrete it that was actually affected by this single suboptimal transient. And in the world of CAR T, that's a genetic construct. So, these are lessons that we have to kind of, and the commercial consequence. Okay, this was a huge commercial consequence that was due to one suboptimal transient. My next example is a publication a year or two before that by one of the most knowledgeable stem cell experts in the world. And they were investigating the loss of the repopulating capability of bone marrow stem cells, the same type of stem cells that are used for transplant, whether they come from mobilized peripheral blood or cord blood samples or bone marrow biopsies. It's those same cells that give life to probably more people than any other cell therapy. And they were looking at what happens to them when they get exposed to air. And what they found was as soon as these cells see their first rise in oxygen from air, they lose a major amount of their life-saving phenotypic character. And if they eliminated that one suboptimal transient, they get 4, 5, 6 times more repopulating capability. Now, in the world of this clinical thing, right now I think a lot of times there's not enough stem cells in cord blood samples to use just one, so they use 2. And in some cases, the time to recovery, varies for some of the patients. But this is a significant thing. But more significant is how fast it occurred. It occurred really fast. Number 2, it was so sensitive that they had to do all the work, all of it, In a hypoxia chamber, I'm going to take issue with that. That's a low oxygen chamber because it's actually probably normoxic for the cells. And it was so sensitive they had to pre-equilibrate all the plastic beforehand. Otherwise, the plastic would, expose them to this oxygen. Think about that. And this isn't new. There's other examples with this, but the lessons here are pretty astounding, and one of them is how are you going to do this with conventional procedures? What else are we doing in the hood, in air? But the examples here, again, one single suboptimal transient, a potency phenotype that's irreversible loss. That's probably one of the most life-saving cells known to man. And was it the progeny or the direct exposure to those cells that was affected? They weren't really clear about that. But one thing they were clear about is it didn't have anything to do with viability at all. And it didn't have a lot to do with anything else they could actually measure. And so, this lesson of what's happening before the cells start to die is very, very, very well represented in this example. And then, the final lesson here is this is a clinical consequence that's still going on every day in every bone marrow transplant facility in the world. My fourth example is, again, oxygen. And this has got a little story to it. This has to do with the discovery of vascular endothelial growth factor. This was discovered in the 90s. It was a factor circulating in the blood that was determined to be the factor that stimulated blood vessels to grow in the middle of solid tumors. And when this was discovered, it was just, everybody wanted to study it. And this VEGF expression was found to be induced by hypoxia. So everybody wanted to do a hypoxia. And this is a story that I can personally relate to because we were designing and selling some of the most popular, effective hypoxia experimentation in the equipment in the world. And what happened at the time for several years was a huge amount of variability and unpredictability in these experiments that were being done in these laboratories all over the world. And in our case, we'd have people say, hey, I bought your equipment to replicate some results I saw in another paper, and it doesn't work. And something's wrong with it. So we did look at the equipment. There was nothing wrong with the equipment. It was doing its job, but they couldn't get the results. In other examples, people would say, gee, we bought your equipment to express this VEGF, and boy, it worked great for 4 or 5 months, and then all of a sudden it doesn't work anymore. Something's wrong with it. So they would have us look at it, we look at it, nothing wrong with the equipment. And this went around for several years, and all of a sudden, people started to realize that this phenotypic expression was turned off by such a short exposure to a rise in oxygen that it was very difficult for people to get reproducible results. And so this actually has started a change in the whole industry for growing and handling cells. And I'll talk about that a little bit more later. But basically, this factor, this hypoxia-inducible factor, is an extremely, fast-reacting transcription factor. Although it takes hours to upregulate, it only takes seconds or minutes to downregulate. And this is in the context of some media inertia, but it shows you how fast the growth factor or the transcription factor itself is responding. It's fast, faster than anybody would believe. And in the point where people realized that they had to avoid this downregulation, the solution for people that had a low oxygen incubator was, this is what we told them, is that, if you want to get this successful phenotype, you have to put your cells in there and you have to keep it closed till the end of the experiment. Because if you open it one time, just one time, it'll turn off the entire phenotype that you desire and you're trying to get. That's how sensitive it is. So, for many years, this was just kind of considered to be a VEGF and some of its cousins situation. And this extreme labile nature of this transcription factor, was widely discovered and known. But what's happened recently is now there's a whole bunch of other phenotypic factors that are just woven throughout all aspects of cell metabolism and cell functions. And these are all partially expressed in the context of this same transcription factor. And now what's happening is a lot of this lack of reproducibility when people want to express mTOR reliably and c-Myc reliably and so on. So, it's expanding into a bigger world than anybody ever anticipated. And I put this quote up here because this kind of makes a point that's important to understand, is that, the entire process of organogenesis, of development, takes place under hypoxic conditions. Again, I take issue with the word hypoxia because he's talking about hypoxia to us, standing in the room breathing air. What he really means is that the entire process of organogenesis unfolds under normoxia for these cells, and it's a really low level. That's normoxia for those cells. But what this kind of means is that the genetic program that is in each of us when we are a zygote basically has a 16 to 20 year run before the program has been completed. Of course, in my case, it was 30 years, but it's a time constant. And it makes sense that Bayer would get a huge negative consequence in their big bioreactors. It doesn't make sense. I mean, nobody knows the mechanism, but this kind of explains how one transient could have a big effect. So, running out of time, aren't I? I would say every suboptimal oxygen transient contributes to phenotypic confusion. I'm going to call phenotypic confusion something that is a little bit unusual, and it results in potency degradation. There's a huge amount of the genome that's affected by oxygen concentration. And the oxygen concentration is active in the epigenetic and in the transcriptional realms of gene expression. So these oxygen-regulated genes are virtually everywhere. And the key thing that's starting to be understood is there's a lot of oxygen enzymes that use oxygen as a substrate, and as we know from Biochem 101, all these reactions have a rate that's a function of the substrate. And there's this new discovery of this huge superfamily of dioxygenases, which have different affinities for oxygen that are distributed in all the cells and all the tissue of the body. And this is explaining now how some of the epigenetic control with histone methylation and demethylation kind of sets the stage for whether you become a B cell or a T cell maybe, or whether you become a heart cell or a liver cell. And this is a fast reaction. And these enzymes are very critical involved in cell fate. So, what's a physiologic level of oxygen? Well, it depends upon which cell. A bone cell might be normal at 1. A lung epithelium might be normal at 12. And the next question is, well, what's pathophysiologic, which the cells want to respond against to? Well, that depends upon what's physiologic. And in every case, what you've got is you've got the enzymes, these dioxygenases with different affinities, monitoring this and making sure that they respond appropriately for every different type of cell in the body. And these enzymes are everywhere. So, there's a big diffusion gradient, a huge amount of infrastructure in the body to get oxygen to where it needs to be. It's a diffusion gradient that comes, through the whole blood system, and the affinity of hemoglobin. And at the capillary, it's a diffusion gradient from the beginning to the end, and that's where it diffuses out into the tissue. And if you take a cross-section of the capillary, you can look at the diffusion gradient that comes out of each capillary due to consumption of that oxygen. And the cells that are the furthest from the capillary have the lowest oxygen level. And it's now pretty certain that this plays a role in the structural formation of different types of cells in the parenchyma of different tissue. And certainly it's well studied and well documented in liver, and the same results are coming out with other types of tissue. So, this set of dioxygenases is everywhere and is sensing it all the time and is responding quickly to fate decisions, to phenotype decisions. And that's why I can fairly be confident saying that our existing dependence upon, just media inertia and cellular homeostasis is probably, causing a lot of handicaps in the potency of our cells. And I want to make another bold statement here too, is that any hyperoxic exposure probably contributes to genotypic toxicity. And there's always a balance between oxidative stress and the antioxidant defenses that cells are naturally endowed with. But the more oxygen there is, the more molecular damage there is. And, air oxygen has been published to be genotoxic since the 50s. There's hundreds of publications that specifically point to the genetic damage caused by air. And of course, that's the suboptimal transient that we're usually exposing ourselves to when we do expose them to one. So, it causes all sorts of genetic damage. And I would normally want to put a plug in for my company, which makes the equipment that avoids these suboptimal transients. But I am running out of time, and I'll spare you the commercial plug. And I will move right to the one point that I want to make before we close here, which is, I think, a significant point. Variability, it's a curse that we are dealing with every day, all the time. The whole society is. In fact, the taxpayers are dealing with our variability. You've heard about the scandalous waste of $20 billion a year and irreproducible results, right? Well, I'm not saying that suboptimal transients are all of that, but I am saying that what has to be done with the concept of variability is you can't just, glump it into one thing. Variability is a multifactorial type of thing, and it's the only way that we can address variability is to look at it as multiple sources and then deliberately address the source of that variability. So I would say absolutely confidently that suboptimal transients are a major cause of this $20 billion worth of waste of taxpayers' money. It's not the only thing, but it's one of those. I guarantee it, because that's what people have been publishing on for a long time. And the big news about, Kymriah, Novartis' amazing cell therapy that's curing childhood cancers. One of the big publicity things that came out after they started commercializing was, oh no, they got manufacturing variability, and headlines were manufacturing problems, but if you read underneath it, you have these pundits who are saying, well, geez, it's different patients, and different patients have different cells, and others say, well, that's the way cells are. But I don't think that's the way that we should address variability. I think we should address variability by dissecting each source out and addressing it, and at least it decreases it. It may not solve everything, but at least it decreases. That's the productive way to go forward with this huge, challenging problem. And I guarantee you, because I know how they manufacture that, and I guarantee you part of their problem is coming from suboptimal transients of these critical process parameters. So thank you for your attention, and hopefully you go to my website and look at our phenomenal equipment.
Yeah, and he also has a booth and he's a sponsor as well. So